Okay, so somebody throw me the anatomy of the heart on this. If you squint at a heart and kind of like tilt it in the right direction, it sort of looks like this. But more realistically, this is approximately what a heart looks like. So that was a joke. You don't have to actually try and label an anatomy this. That would be rather difficult, although I'd be interested to see what people would try to do. But let's just go ahead and go through the anatomy real quick. So I'm going to have the people who are more senior in the lab go last so that they end up with the harder things, hopefully. So does anybody want to volunteer to label one structure on the heart? And if you're on your phone, you can tell me what you want to label so I can write it for you, because that looks like it would be annoying.
I can go.
So the aorta. What is the aorta? Can you describe it a little bit?
Me?
Yeah.
It is the first and the biggest artery which leaves the heart and takes oxygenated blood.
Speak big.
Sorry. It is the first and the largest artery. First meaning first to leave from the pump system of the heart. And yeah, all other arteries branch off from it.
Okay, cool. That's a good start. Okay, who wants to go next?
Okay, I'm Eric for people that don't know me. I'll do the superior vena cava up there.
Describe to everyone what is the function of superior vena cava?
It returns blood to the atrium, mainly from the upper extremities, like the brain. Is that it?
Yep. And superior vena cava, so superior meaning top, right? So you see it's on the top of the heart. And then vena, so it's a vein, it's part of the venous system. And then cava, cavernous, it's the big vein. It's the high big vein. Okay. Who's next? Okay, so right ventricle. Okay, so what part of this are you saying is the right ventricle? Is it that black line?
Yeah, the
right,
top part.
right there? Okay. So if I was to draw a circle around the right ventricle, what would it entail? Like what parts would it cut off? So is this right ventricle?
No, so the ventricle is going to be like around this area.
Yeah. Okay. Good. What is the function of the right ventricle?
It's going to contract to push blood back up through this valve right here.
And where does the blood from the right ventricle go?
That will return.
Everyone, feel free to help out too.
It goes to the lungs. Right?
Yeah.
Yeah, it goes to the lungs. Okay, so what kind of blood is it?
Deoxygen.
Deoxygenated. Okay, good. Okay, Derek.
I don't know if I'll be able to write it.
Yeah, you can just point. What do you want to label?
Maybe right below the aorta, the pulmonary artery.
This thing?
That thing.
Okay. That's the pulmonary artery. Okay, and what is the function of the pulmonary artery?
It's the path from the right ventricle to the lungs.
And so, what's the difference between an artery and a vein? Anybody?
Artery goes away from the heart, typically, and it goes towards the.
Yeah, no, it's exactly that. Say that louder.
Oh, sorry, I keep forgetting. Arteries go away from the heart, veins go towards the heart.
Okay, and so it's not necessarily about if the blood's oxygenated or deoxygenated, right? That's sometimes a misconception. But here we have the pulmonary artery taking what kind of blood?
Deoxygen.
Deoxygenated blood away from the heart, right, to the lungs. Okay, so it's about which direction it's going with respect to the heart. Okay, Ben, label something.
I'm going to label the tricuspid valve.
Wonder why.
I don't know.
For reference, Ben's currently working on a paper that involves the tricuspid valve. And while you're doing that, tell us why it's called the tricuspid valve and what it's between.
It's called the tricuspid valve because it has 3 leaflets, which are like the little, I don't know, veins that come together to like form the valve. And what was the second part of the question?
What does it do?
Oh, it prevents blood from flowing back into the atria when the ventricle contracts.
Okay, so it's between what and what?
The right atria and right ventricle. I'm sorry, I'm trying to draw
There you go.
the line.
Okay, cool. Thank you, Ben. And then Anna, why don't you draw a thing or label something for us? Anna wrote. Did you write chordae tendineae, Anna?
Yes, and is the arrow is being kind of weird.
Yep. No. Yeah, I see it. Y'all see it? There. Okay. What is a chordae tendineae? Anybody here know what that is? Heard that before?
Yeah.
Yeah, I know you probably know. Yeah.
Yeah,
Go ahead.
It's like it connects the valve to the wall of the ventricle to hold them so that they don't invert.
Yeah. Yeah. Awesome. And then let's go ahead and do Lindsay. Will you label something?
Right here, you have the pulmonary veins.
Okay, what do the pulmonary veins do?
They bring oxygenated blood from the lungs to the left atria.
Okay. And so that means that this thing that they're connected to right here is the left atria, right? And then I'm just going to fill in a couple more and I'll let James tackle a harder question at the very end. So left atria. So let's start here. Blood comes from the left atria, then it goes where? Atrium. Or atrium. Left atrium. Atria is plural.
Left ventricle.
Goes to the left ventricle, so that's this chamber here. Okay, LV. Okay, and how does it get there?
Mitral valve.
The mitral valve. Okay, so that's this one right here. How many leaflets does the mitral valve have?
2.
2. Okay. Blood goes from the left ventricle where? Lindsay, undergrad?
Back through the aorta.
Yeah, to the aorta. Through what? This one's simpler than it seems. So what kind of thing does it have to pass through?
Oh, no.
Yeah, and what is the name of this valve between the ventricle and the aorta? The simple name.
I'm sorry.
Anybody? Aortic valve. Aortic valve. Okay. Okay, then the blood goes to the aorta, to all the body, right? And then it comes back to where? Derek?
So you said it goes out of the aorta and then
It goes to the body, and then how does it get back?
through the superior vena cava or the vena cava.
Yeah, the
veins.
vena cava. Yeah, so superior, right? That's the one Eric showed on the top, and we also have the inferior, IVC, on the bottom, right? Inferior just lower. Okay, and that drains into where?
The right atrium.
The right atrium. Okay, right atrium goes where?
Tricuspid valve to the right ventricle.
Through the tricuspid valve to the right ventricle, and then Eric, bring us home.
Through the mitral valve to the pulmonary artery.
Through the pulmonary valve.
Pulmonary valve, sorry.
Yep. And then that goes to the lungs, and we come back to the pulmonary or veins, and cycle continues. Okay. Okay. I know that we spent a lot of time on that, but anatomy is important. Okay. And what is the primary function of the heart? What?
Pumps blood.
Pumps blood. Anybody else? Okay, pumps blood is what you'd say if you were a doctor, but the primary function of the heart is to produce an electrical signal. Because that's the interesting part. And it also happens to pump blood or whatever. Okay, so are any of you particularly squeamish? Okay, so I'm going to show you a picture of a real heart from one of our experiments. Oh, let me clear my annotations here. Thank you all for participating. So this is an example of a heart from one of our experiments. And so it's a little bit harder to identify anatomy here, but we can still try, right? So ignore this weird plasticky thing. What is this top section here, do you think? You just take a guess. And we can't see the whole heart right now. It's a little obscured, but anybody have an idea what they think this is?
Right ventricle.
Yeah,
Yeah.
that's the right ventricle, right? So now let's go ahead and orient ourselves. We're laying on the table, okay? So that's the right side, okay? Another reason you can tell it's the right ventricle is it's a little hard to tell if you're not used to looking at this, but you see there's this division right here where this vein is coming down. Okay, that's known as the left anterior descending coronary artery, and that kind of splits the left from the right ventricle. So this whole region down here that's larger, what must that be?
Left ventricle.
Left ventricle, okay? And so this is going to be thicker, more muscular, and larger overall, and then the right ventricle is kind of like a flap hanging out the side of it. Okay. Okay. If we have time at the end, I'll show you some videos from one of these experiments of the heart actually beating, but I want to make sure we have time to get through the other stuff. Okay, so now we're going to talk about the primary function of the heart, which is its electrical activity. And we're going to do kind of a similar thing, but maybe hopefully a little bit more rapid fire. Where does electrical activity in a normal, healthy heartbeat start? The sinoatrial node. Okay, everybody hear that? Okay.
Sorry, I'll talk louder.
No, you're good. Where is that?
On the sinus of the atria.
Someone circle it. Circle the approximate region in this. Okay, yep. So that's the sinoatrial node. Okay, so then where does the activation go next? What is the next tissue to get activated? Go ahead, Eric.
Atrium tissue.
Yeah, the atrial tissue, right? So we have a wave of activity spreading throughout the atria, right? And you'll hear Lindsay talk about this a little bit in the next project for the newer undergrads. It's kind of like you imagine you drop a rock in some water and you see the ripples move out from it, right? So if we drop a rock right at the sinoatrial node, then we see a ripple of activation spread across the tissue. Okay. So what's the next important event? Ben, what's the next important place that activation is going to go to?
The AV node.
Okay, the AV node. And what does that stand for?
Oh, I don't know. I don't know what the A stands for, but it's something ventricular.
Okay, so what is it between?
Oh, the atria and the ventricles.
So what does it stand for?
Atrioventricular node. Is that it?
Atrioventricular node.
Atrio.
Yeah.
Okay.
So what is the function of the AV node then?
It kind of doesn't it delay the signal slightly prior to letting it continue to the ventricles?
Yes, so that's one of its functions. So the atrioventricular node takes the signal that's from the activating wavefront in the atria and it slows it down. But then it also does an important thing in that it's the only, in a healthy heart, the only bridge between the atria and the ventricles where electrical activity can move. Okay, so everywhere else where you see the divide between atria and ventricles, there's a fibrous tissue there that's non-conductive. Okay. Okay, so then Lindsay, grad student, what is the activation? What comes next in the activation sequence?
It's gonna go to the His bundle.
Okay, and can you just kind of describe what the His bundle is a little bit?
Is a bundle. I don't have a better definition than that. Just kind of is a little breaking point, and then it'll just like a little short wire, and then it travels from there and it branches to cover the rest of the heart.
Right. And so the important point I want to point out there is it's like a wire, right? In a sense that it allows conduction to happen very fast. Okay. So because the thing is like as your heart is contracting, part of the whole function of this electrical system is to get your heart to contract synchronously in a concerted manner, because as your heart contracts, it needs to pump blood. And the best way to pump blood is to have the chambers that are doing the pumping do so all at once, right? But you can imagine that might be a problem if it takes some time for the activation to spread over the whole heart. Okay, so the heart essentially has these kind of wires in it that take that signal and spread it really fast. Okay, and the His bundle, right after the AV node is what's going to essentially be the starting point for that activation to spread to the whole ventricles.
It's also worth noting probably that in the body, when we refer to like electricity and wires, we also refer to nerves sometimes, but that the electrical conduction system of the heart are not neurons or nerves, but rather electrically active like cells rather than like nerves, which anyway, that can be confusing for people sometimes. So worth noting.
Yeah. And it's also worth noting is that another distinction between neuronal and cardiac is that neuronal activation spreads via neurotransmitters, right? One neuron will activate and then it'll stimulate the activation of another one. But with cardiac cells, a lot of them are directly electrically coupled to each other. Okay. Okay. So does anybody know what comes after the bundle of His? This is a little bit more detailed than you might have learned, but
Purkinje fibers.
Purkinje fibers. So you actually have, right before that, you have a left and a right bundle branch. And that's essentially this wire bifurcates into, this side's going to go to the left side of the heart, this side's going to go to the right side of the heart. And then it branches out into what are called Purkinje fibers, which are like a net of wires that covers the entire ventricles. And this is so that that electrical activity can very rapidly be brought to the whole ventricles at once so that they can contract pretty much all at once. Okay? So that's kind of the approximate activation sequence of the heart. And we're just going to walk through what that looks like in these schematics here coming out. Does anybody have any questions so far? Rather, who has questions at this point? Don't make me go one by one.
Are there significant differences or to the right and the left close exactly at the same time, or are there any significant differences between when they close, or not close, sorry, contract the ventricles?
Yeah, good question. Depends on how close of a magnifying glass you get on things, right? So the electrical activity spreads mostly evenly, but it's not completely even, as we're going to see in a few slides here, right? And so there's certain parts of the heart that see the signal a little bit later than others on a very small time scale. And typically, because the right ventricle is so much smaller than the left ventricle, right, it's easier for that wave to get all the way through the right ventricle, where the left ventricle will still have a little bit left over, not quite gotten to it yet. But from a macroscopic view, looking at like total heart contracture, it for the most part looks like the ventricles are beating together, right? And it might be that like it's a little bit, the right side starts to tighten as the left side's starting to, is not quite there yet. But for the most part, it's conservative. And granted, this is all happening on the order of, tens, 20s of milliseconds.
From a pathology perspective, if they're quite uncoupled, it might indicate alterations in signal propagation, which could indicate some kind of, pathology or block or whatever.
Yeah, and that's where it gets very interesting. And that's kind of Lindsay's going to give a talk about this at some point of trying to understand, those sorts of, can we diagnose those sorts of things from the measurements that we take. Okay, thank you. So just to recap, we have the activation starting up here in the high atria by the Sinoatrial Node, right? And this is going to give, these activations are going to give rise to an electrical signature that we can actually measure on the body surface. And so that's what you're going to see traced out over on the side here, right? So the atria contract or activate, and when we see that electrically, we call that the P wave. These are just named semi-alphabetically starting at P. The next part is that the atria contract completely, and then there's a little bit of a delay, and you can kind of see the signal, the purple is spreading into the His bundle and the bundle branches, right? So it's not quite activating the ventricular tissue yet, maybe a little bit, like right here in the septum, but not in a way that we'll notice it. So as far as we can tell from the body surface, from our electrical perspective, there's no electrical activity. So we see a flat line on the electrical recordings. And it's also during this time that the atria contracts, right? So there's a little bit of delay between the activation and contraction, right? So does anybody know the name of the wave that comes next? I told you it's alphabetical, so you should be able to guess. X. What comes after P? Q. Okay, so the Q wave, right? And from the perspective of this electrode, it looks like it's negative. We'll talk about that a little bit in a second. But that's as we start to get the septum activated, right? And we can see that this activation wavefront is racing down the Purkinje fiber, or the bundles of His, and starting to go into the Purkinje fibers. Okay, and then we get the R wave, and this is where the bulk of the myocardial tissue, the ventricular tissue, starts to activate, and right, so we see a really big spike. We see a lot of activity all at once. Okay, and then after that comes the S wave, right? So QRS, and this whole complex, this QRS complex, as it's called, represents the activation, the electrical activation of the ventricles. Okay, and so it mostly happens at the same time, but you can see there is a delay between these different waves. So certain parts of the heart do see the activation before the others. And typically, the part that activates first is also going to contract first. Okay, it gets a little complicated, and there's a little hysteresis to all of this, but okay. So shortly after the QRS complex, the ventricles contract, and this is a little schematic. The timings here aren't like perfectly laid out. Okay, and then after that, you have a period of no activity, right? And this is called the ST segment, okay? And then something we're not going to get into very much today, but the way that these electrical activities are passing between heart cells is known as an action potential. That should sound somewhat familiar to all of you, right? And they have a characteristic shape, which involves the electrical potential of the cell going up and down, right? So in this case, as the heart cells all activate, their electrical potentials go up, but then eventually they have to come back down to normal. And the phenomena of all of that of all the cells in the heart coming back down to normal gives rise to another electrical wave. It's not quite a wave, it's more like a thing that happens about the same time throughout the heart. It doesn't propagate from cell to cell quite the same. But that gives rise to another characteristic wave on the ECG, which is the T wave. Okay? So then the total activation of the heart, right, atria and ventricles, will look something like this on any electrode that you measure it on if it's far enough away. Okay? Who has questions about this? I just glazed over like 2 courses worth of material.
I have a question.
Mm-hmm.
First, can you remind me of the name of the node that like it starts with?
Okay. It's a sinoatrial.
The sinoatrial. So when that activates and it goes across like the atria, is it similar where it has like those like connective like conductive fibers, or is it kind of just like running through the cells? Like they did say that like they're not necessarily nerves, it is like conductivity of cells themselves, but is it similar to how the ventricles are running?
That is an excellent question. Matter of hot debate. So there are theorized to be similar Purkinje-like structures known as internodal tracts, which would be between the 2 nodes, the SA node and the AV node. And there is competing literature evidence on if there is or is not. I think what's currently in vogue is that there's not.
Like there's nothing as specialized as the Purkinje system, because if you go on the cellular level, if we showed you a Purkinje cell versus a working myocardial cell under a microscope, you'd see a profound difference, even for a non-specialist. They have a different structure. Purkinje cells, for example, don't contract. And yet virtually every cell in the atria does contract, has contractile proteins. So there are no highly specialized conduction system cells in the atria that we know about, but there are bundles. And so there are bundles of otherwise more or less normal myocardial cells that are sort of formed together into groups that have a consistent orientation. And because they have the same orientation, they tend to conduct a little more rapidly and form these links between different parts. So you'll definitely hear different structures. I don't want to get into them, but you'll hear of different structures in the atria. They're muscular structures, and they're thought to play some role in the spread of activation. But as Jake said, there's lots of debate about it. But there's no equivalent to Purkinje cells. That's the important part that we do know.
So is that another reason like that we could believe that like the specialized cells are going to cause like a way higher like electrical wave just because of like how they carry versus like just another cell?
It's actually not quite that. So the magnitude of the electrical activity that we're able to measure is more due to the bulk of the tissue, right? So the Purkinje fibers and all that, they're very, very small amount of tissue, right? And they move really fast. So 1, you have to sample it pretty fast in order to find it. But 2, it's a tiny amount of tissue in an ocean of other activating tissue, right? And it just generates very small electric fields that are really hard to see. Whereas when you have in the ventricles, right, you have all of the ventricles starting to activate at once, right? You have this big wave front. And that's a lot easier to see because it's all moving in the same direction.
Okay.
But when you said higher, what? Well, your question said something that the Purkinje cells produce something higher, but you didn't say what it was that's higher.
Right.
Oh, I was going to
Jake's
like
talking about amplitude of the resulting signal in the ECG, but that may not be what you did.
Yeah, I was just mean like I guess kind of like I was wondering if like the voltage across them would be the same, like if we're actually measuring like a similar.
They have a different shape of action potential, but a very similar magnitude of action.
Okay.
But they do have a different shape. And like I say, they don't contract. They're tightly coupled cell to cell. And the net result is that the signal moves through that highway. You can picture really, it's like, and it's a similar sort of thing. When you think of the whole landscape of Utah, there's a few highways. And they carry a disproportionate amount of the traffic, and they're clearly the fastest way to get through the state. And that's what the Purkinje system is like. It's a specialized pathway made of, very different, like the highway is a very different landscape from our desert.
Mm-hmm.
And the Purkinje cells are similarly different from the working myocardial cells.
Okay, thank you.
Have any more questions? We're gonna move on. Because what we're gonna move on to is kind of built on understanding how this activating wavefront kind of goes. I've glossed over a lot of cellular details. We can always talk about them later, but okay. Cool. So Ben will remember this exercise that we're about to do. So there's various ways we can think about this electrical activity, various ways we can represent it, both conceptually and mathematically. Okay. And what we always have to keep in mind is that every model is wrong, but some models are useful, right? And we have to know what they're useful for and all that other good stuff. So I told you that, and we saw it in this diagram, that this kind of activating wavefront, it moves around the heart in a semi-ordered fashion, and during a healthy heartbeat, it'll move kind of in the same pattern over and over again. Okay. If we want to view that electrically, right, we have essentially on the cellular level a bunch of different charged particles. On one side of the wavefront, there's a certain charge, and on the other side of the wavefront, there's a different charge. Right, and that gives rise to a bunch of little dipoles, right? Charge differences between two places, okay? And so if we take those dipoles and we sum them all up, we can get a single dipole that represents the activity of the heart at any given time point. Okay. Am I losing anybody here? This is going to be important going forward. Please ask clarifying questions. This includes everyone. Okay. So, for instance, I'll go ahead and draw what a couple of those maybe would look like, right? Maybe that'll help. So let's see, let's insert a arrow. Okay, so and I'm just going to center them all on the center of the heart, right? Because this is like we're looking far enough away. We're saying what is the approximate direction of activation, right? Is what we're representing by this dipole. And so we can see here, we've got, kind of the electrical activity starting up in the SA node, and it's moving in kind of this downward direction, right? So all those little dipoles on that edge are going to be mostly pointing that way, right? Crappy color for this. Does that make sense, everybody? So we got hundreds, millions, infinitesimal, infinitely many little dipoles pointing in that direction. Right, and so we're going to represent that total activity by a single dipole vector pointing about this way. Okay, get a little bigger so I can see it. Okay, so then now when we go down to the Q wave, right, rather than pointing kind of off angle like this, it's mostly pointing down a little bit towards the end of the P wave. Okay, that makes sense. Okay, and then during this PQ segment, there's not really anything noticeably activated. Right, the Purkinje cells and whatnot, and the His bundle, these like specialized highways, they're activating. And if we got in close enough, we could see little dipoles pointing down them. But usually, when we're far enough away, we can't really see those, and so the vector goes away completely. We don't see anything. Okay. Then we get into the Q wave. Okay, and we can kind of see this one's pointing, kind of straight downish. Right, it's activating down the septum. That makes sense. Okay, now we've got the R wave. Okay, this one's a little complicated, right? The septum's already, for the most part, pretty activated. So now we're activating kind of up and around the sides of the ventricles. And so we kind of have to think about this idea of superposition. We've got a bunch of different vectors, but when they all add together, what's their overall direction? Okay. And in this case, it looks like maybe more of them are pointing kind of towards the left a little bit. Okay. And then eventually, as we get a little bit farther, they're going to start pointing kind of up, right? As we start activating up the ventricles, right? So we're looking through a slice here, so it's kind of hard to see, but we've come down the septum. Now we're going up the walls of the ventricles. Okay, and then once again, we're going to get kind of towards the end of the QRS here. And it's going to still be mostly up, right? That's mostly what's left to activate. And I'll just tell you from intuition that the left ventricle is a little bit bigger, right? And so it's going to be skewed a little bit towards the left ventricle because that's going to be where the last little bit of tissue is to activate. Okay, so we've got these series of electrical dipoles, right, that are representing the heart. Does that make sense? Okay. And then I'm not going to talk about the T-wave at all because it's very complicated and it's not well understood. So then we want to figure out a way to actually measure this and have some intuition for what we're measuring and what it means with respect to the heart, right? So if we're saying we're going to represent the activity of the heart with this dipole, then when we measure characteristics of this dipole, we can then intuit certain things, like this is the direction activation is going broadly in the heart. Okay, does it make sense how that would be useful? Okay, so in order to do those, we need to have a certain number of electrodes, right? So let's say that these are 2 electrodes and they're arbitrarily far away from the heart. Let's say I just put 2 electrodes right on your chest surface, right? And I'm going to measure the potential between the 2 of them, the electrical potential between the 2 of them. Okay, so what is that potential going to look like if we have a dipole that let's say that dipole looks something like this. Make it bigger so everybody can see it. Okay, so, and just as a note of some conventions, the arrow side of a dipole is usually represented as positive. Oh gosh, instead of doing that. So the arrow side is usually positive, and the base side is usually negative. Okay, and let's say for the purposes of our experiment, we have our voltmeter here, right? We've got a positive terminal and a negative terminal, and I'm going to hook them up like this. I'm going to cross this one over here, right? I'm going to cross this one over here. Okay, so this is the positive terminal of my measurement, and this is the negative terminal of my measurement. What is this going to read out? Not in absolute terms, just what kind of voltage? Positive voltage, negative voltage, 0 voltage. Positive voltage. Why are you saying positive voltage?
Yeah, the positive side of the dipole connected to the positive end of the meter.
Okay.
And it's gonna be, the arrow is positive, there's going to be more positive built on that side.
Right, so let's think about it. So when we have a dipole here, we can also think about the electric field generated by that dipole, right? So we've got a bunch of positive on this side, a bunch of negative on this side, right? And that kind of, that field radiates out away from the dipole. Okay, and so when we go to make our measurement, we're doing our positive side minus our negative side. So if the positive side has a higher potential and the negative side has a lower potential, then it's be high minus low. That gives us a positive number. That makes sense to everybody? Okay, and what if I flip my leads around? Negative but equal magnitude. Okay, that's very clear to everybody? Okay, cool. Now what if instead of that dipole, I had a dipole that looked like this? Right, so this is maybe, somewhere during the QRS, right? We've got the activation mostly going up. And I'm measuring it with these 2 electrodes right here. What am I going to see?
0.
0. Okay. So someone walk me through how you came to that conclusion.
I mean, like if you want to look at it physically, it's because
whatever makes sense.
Okay. Well, I mean, using linear algebra, it's orthogonal to each other, right? Like the vector, the dot product of the 2 vectors is 0, so it's going to be 0. If you're going to look at it physically, it's just that it's measuring at the same electrical, or it's measuring at the same voltage, and so it's going to be same minus same, which is 0.
Right. So think about it physically first. Like you said, if we draw out kind of the electric field lines from this, we should see that these 2 points are going to be at the same potential, same minus same, 0. Okay, but this other idea he brought up, this linear algebra one, does that make sense to everybody? What he was getting at? Right, we can think about this measurement here as another vector, right? And what are we doing to our dipole, our source vector, in order to get the measurement? What is the process that you can think of?
We're projecting it onto it, right?
Right, we're projecting it. Okay, that's very important. Who has a question about that? Because that didn't make sense to me when I first learned it. Granted, I didn't have a math background. Anybody have a question? Okay. So, do you think then, given that we kind of walked through all these different vectors for the activation of the heart in this simple case, do you think 2 electrodes is enough? How many electrodes do you think we would need?
Depends on what you're trying to find.
Explain.
Well, like if you're trying to take a really robust, like you can get some idea of electrical signal propagation with 2 electrodes, right? Like you can have an idea if the axis of signal propagation is like super off. But, if you had more angles, in theory, you could look more closely at different propagation in different directions. And so that might point you towards different pathologies that you might miss if you didn't have as many angles as you do looking at the heart.
Okay, that was a great answer from James, and hopefully it reminds you of those trick questions I asked some of you when I was your TA in Fundies of whenever I ask you a question about is this enough, you always have to say, well, it depends. What's your question? Right? What's the thing you want to do? So like James says, if you really want to investigate the nuances of some particular pathology, then you might need a lot of electrodes. So let me refine my question a little bit. How many electrodes do we need to characterize all these possible vectors that we showed here?
2 dimensions or 3 dimensions?
Good question. Good clarifying. Let's just stick to 2 for now.
Another set.
Another set?
Yeah,
So
another set of 2, so 4 total.
So 4 total. Okay. Could we get away with any less than that?
Yeah, you could do 3, right? It would just
Why?
be more complicated math because you would still have then a vertical component. Or if you did between the 3, if you measured between the 3, I guess, would that be a tripolar system? I don't know what you would call it.
So can anybody tell what he's doing in his head right now? Why is it important? Like the arrangement of these electrodes is important. So what are we missing with just these 2 electrodes?
Vertical component.
Right, there's no vertical component. So if ever the dipole is like whenever the dipole goes up out of view, right, we can maybe assume how much of it is in the up direction, but we don't know. Okay, and if the dipole is straight up, we can't tell if the heart is quiescent, not doing anything, or if it's doing a lot of something in a direction that we're not measuring. Okay, so we need some intuition of the vertical component. Okay, and so like he said, we could get away with 3 electrodes, right? Because we could put 2 on the horizontal, and then we could put 1 on the vertical, right? And that would give us, reference points. But the math is a little bit more complicated; it's less intuitive. So let's just work with 4 for now. Here are my annotations. I think. Okay, so we have 4 electrodes here. How do we want to hook them up? What do we want to be positive? What do we want to be negative? This is the choice. We get to make this choice.
Stick with conventions and do the top and the right are positive, and bottom and the left are negative.
Okay. So positive, positive, negative, negative. Is that what you're saying?
Mm-hmm.
And then what are our pairs here, or tries, or what, like how are we hooking up? So how many measurements are we making right now? How many measurements do you want to make?
We're probably gonna go with simplicity. Let's just do 2. So you left and right are a pair, top and bottom are a pair.
All right. So we'll call this pair number 1. We'll call this pair number 2. Everybody on board with those conventions? Okay. Doesn't matter what conventions you pick. I mean, it matters in some senses. Some will make your life easier or harder. Just pick conventions and stick to them and have a reason, right? So our reason here is this follows the standard XY Cartesian conventions, then it's easy to think about. If we're investigating a certain disease that always makes things point down, then maybe we want to reorient everything so our numbers are positive. Who knows? Okay, so let's go ahead and take these and take this small smattering of vectors that I have here. And we're going to go kind of from red to pink. So red, orange, green, blue, purple, pink. And we're going to kind of figure out what the approximate potential is going to be for each of these. There's no numbers associated with them, obviously. We're just going to kind of do relative values to each other. So let's see. So let's start with lead number 1. So, and let's assume everything starts at 0, right? So before red, there's no vector. So everything's going to start at 0. Okay. So what is the potential going to be for the red vector on lead 1? First off, is it positive or negative?
Positive.
Why?
It's the same as the one we were talking about at first. So the same direction. Positive on the right.
All right, so if we draw our imaginary lead vector, right, with a tail here and a head here, right, and we take the dot product between this vector and the red vector, it's going to be a positive number, right? Or to think about that another way, if we project this vector onto the black lead vector, right, it's going to give us a positive magnitude. Okay, everybody clear on that? Very important. Okay. So it's going to be some sort of positive value in the beginning. All right, let me try to make it red. Okay. And then what about when we go to orange? Still positive, still negative, or goes to negative? What do we think?
For lead 1.
What?
For lead 1, sorry.
Yeah, for lead 1. So Lindsay, you're nodding yes, still positive. Higher positive or lower positive? More or less positive?
Higher,
Okay. So and why do you say that?
because it's more in the direction, in the horizontal direction.
Right. So the magnitude of after projection is higher than it was during the red. Right. So if you think about taking this orange vector and projecting it up, right, as a longer projection than when we did the red one. So it's going to go up. Okay. And then what about the green one? So Eric, what is the magnitude going to be on the green?
Roughly 0.
Yeah, maybe a little positive,
A little bit, yeah.
but it's going to be pretty low here. Okay, Ben, what's it going to be for the cyan vector? Also, I'm sorry if anyone's colorblind. I tried.
It's going to be a small negative.
Small negative. That look good?
Yeah.
Okay, Anna, what's it going to be for the purple vector on lead 1?
More negative.
More negative. Okay, and then Lindsay, what's it going to be for the pink one?
I think she's dealing with the dogs.
Oh, okay. Oh, geez. James, what's it going to be for the pink one?
In lead 1,
Yes, lead 1.
I'm going to say negative.
Negative. Large or small compared to like purple and
Small.
small. Okay. So then, right, we can kind of see how this traces out a signal, right? And it looks maybe kind of QRS-like. I might have put the spacing out too far. Does everybody see that?
Yeah.
What's going on here? So just to solidify it, we're going to do it one more time for lead 2 and see how they're different. I think so. I think it's pretty cool. Does anybody know what this collection of vectors is called? A vector cardiogram. Okay, so let's just really quickly, let's blitz through. I'm just going to do it in one color for lead 2, right? And so remember, that's positive top, negative bottom. So red vector, positive or negative?
Negative.
Negative, small negative perhaps. Okay, orange vector, positive or negative? Bigger negative. Green vector, biggest negative. Cyan vector, yeah, smallest negative, let's say. Purple,
About 0. Yeah.
practically 0. Okay, pink,
Slight positive.
slight positive, and then we assume it goes back down to 0. Right? So this is the same heartbeat, right? But look how different the signals look. Right? And that all depends on your vector. But given this, given all of these signals that you see here, you should be able to remake every single one of those vectors. Anybody have an idea how they would do that? Hope so, because we're about to try it. Okay, so I'm just going to get rid of the heart here, right? So what would the vector look like that has on lead 1, which is, oh, I forgot to actually change these. So let's see, lead 1 is the green one, and lead 2 is the blue one, right? That follows what we said before. Okay, so what is our vector going to look like that's in the middle there?
Up and to the left.
Okay, so let's do it one component at a time. I think you're probably right, but let's just do it one component at a time so that we get an intuition for this. So for lead one, what direction is this facing? On this lead one axis?
To the left.
Facing to the left. Okay, it has magnitude 1, so we'll say that's magnitude 1. Okay, and then for lead 2, what is it doing? It's going up and it has magnitude 2, so we need to make it twice as long. Right? And so then if we add these two vectors together, we should get our total vector, which is going to go kind of 1 in this direction. Right, it's going to go out 1 and then up 2, so it's going to be about like that. That make sense? Okay, you'd be able to do that if I give you a signal at this time point. Here's the potential between this lead, and you can make this vector. Okay. I know we're almost out of time. Does anybody have any questions? I've got a couple more examples. Oh yeah, here's the answer.
Is it really useful to have the third dimension when you're doing stuff in the heart?
Maybe.
Maybe we don't know.
Maybe
Okay.
if you want to know. So typically we think of like the most common types, and James, correct me if I go way off base. The most common types of like heart conduction issues are left versus right side. But you know, if you want to like, I could think of a scenario where you maybe have a re-entrant circuit that's, you know, kind of going around the front to back of the heart, right? And in theory, a vector cardiogram that had the third dimension would show an oscillating vector going back to front, back to front, back to front.
The way that we use the third dimension clinically is by using precordial leads. So we're talking about leads that are essentially Einthoven's triangle. So like these leads, but precordials come across this direction, and there's also posterior leads that you can use, and those can help clarify like localizations of different infarctions and stuff like that.
And this vector cardiogram, or VCG, is still used mostly as a research tool or in clinical studies that are related to research. And it, for whatever reason, has always had more of a following in Europe than in North America. So if you go looking for vector cardiogram or VCG, you know, do a literature search on it. The majority of hits you'll get will probably from European labs. Or for whatever reason, it's like all things, right?
Convention.
The fashion is different here from Europe.
Yeah.
Lots of things are different. Cars are different. So it's ECG. Why not?
Got it.
So yeah. So
Rob, I see you have to probably head off after this. Do you have any commentary you want to add in our last couple minutes?
no, this is great. You're all doing really well in terms of following along with this. This is the foundation. So it's really good to get sort of an idea. And if questions come up later, like you think about an hour from now, and you think, well, wait, what did Jake do there? And please get back to us. Because getting a handle on this will help enormously when you get into the research and we start asking you to do things and showing you signals, starting to describe the research we do. So do try and get this, at least reasonably under control. Knowing that, as Jake said, there are tons and tons of details, and we'll get to those as we need, but it's good to have this high level view to start with.
This should hopefully gives you some sort of intuition on how those signals come about and what they may be.
Yeah.
Okay. All right, everyone. I think I have two more examples if we want to walk through them if anybody's interested just to practice, but we don't have to. Does anybody have any questions that they'd like to fill this time with? Who has questions? I've heard Lindsay ask questions. I've heard Tanner ask questions. Derek, you ask questions?
So the convention, like the arrow, the positive and negative side, like, is that just a choice, or is there a reason physiologically we pick positive down here and negative?
Oh, well, for dipole vectors, that's just the convention of how you draw a dipole vector.
Okay.
For how the heart actually works on a cellular level, the extracellular space, which is what we're measuring whenever we measure things, is more negative on the activated tissue side and more positive on the non-activated tissue side. So the direction of the dipole points in the direction that the tissue is going to be activated, right? So activated tissue has a more negative extracellular potential, and non-activated tissue, or the tissue that's not been activated yet, has a more positive potential. And that's why it very conveniently fits that the dipole points in that direction. If physiology happened to be flipped, then we'd always just have to think about a dipole that was constantly scooting backwards. rather than pushing forwards.